All right, so I just wanted to go ahead and go over a little bit of our some of discussion wise on cell parts and cell theory. So you're going to go to your workspace, um, and you'll get to this area here where you can pull up your notes. Through this, we'll walk through each one of the cell parts again, kind of explaining what's going on. So we'll just start here with the nucleus, big central mass in the middle of the cell, usually brain of the cell. It's surrounded by a double membrane called the nuclear envelope or the nuclear membrane. And it's where the DNA is located in a eukaryotic cell. Remember, the big differences between a eukaryotic cell and a prokaryotic cell is a eukaryotic cell has membrane-bound organelles and also the presence of a nucleus, whereas the prokaryotic cell is pretty simple and does not have that. The nucleolus is also located inside the nucleus. It's the site of ribosome production and RNA, and it is found both in animal and plant cells. The next one we're going to kind of tag together is the endoplasmic reticulum. Again, there is a rough and there is a smooth. It is a folded, interconnected sac of channels. It's the site of lipid protein synthesis and lipid synthesis. And remember, our ribosome's major function in the cell is to produce proteins. So the rough endoplasmic reticulum that's covered with the proteins, or covered with the ribosomes, excuse me, is responsible for protein synthesis and movement, whereas the smooth endoplasmic reticulum does not have ribosomes. So it is the lipid synthesis. Again, it is found in both animal and plant cells. The Golgi apparatus is a flattened stack of membranes. It modifies and sorts packages and packages proteins into vesicles, and it is used for either accepting incoming transport vesicles or getting vesicles ready to be sent out of the cell. When we talk about endo and exocytosis, we talk about vesicle intake and vesicle sending out of the cell. So if we could understand that this actual cell organelle is responsible for that movement. It's found in both animal and plant cells. The mitochondria, right there, says energy generator. It is responsible for the cellular energy that we have in our cells, ATP. That's where it's converted through the process of cellular respiration. As you can see, it is very convoluted inside, has many folds where there's opportunities for that energy process to take place. It will convert our fuel particles, our glucose, into that ATP that we need. Has a lot of surface area. So the mitochondria is located both in the animal and plant cells and very important in energy creation. The chloroplast, only found in the plant cells, captures light energy from the sun or from any kind of light source, and is going to convert it into chemical energy through the process that we call photosynthesis. This is how the plants make their food, with the presence of water and carbon dioxide also. And as you can see inside, they have disc-shaped compartments that are stacked on top of each other for the light-dependent reactants. They're called thylakoids, and they're kind of like little power packs that generate the food for the plant. And the chlorophyll is what gives it its pigment or its color. The cytoskeleton, found in both, is a supporting network, both animal and plant, is a supporting network made up of protein fibers, and basically it forms a structure for the cell and anchor points for the organelles to hang on to. Your centrioles, only found in animal cells. It's made up of microtubules, and its major job is to help with cell division in the animal cell. When we talk about mitosis, meiosis, we'll get more into that. The cilia and flagella. Cilia is a short, and it's usually numerous projections. Kind of looks like hair on the outside of a cell, where the flagella is longer. It tends to be less numerous, but it's a whip-like structure. They are both um, or what we call motility, helps with movement of the cell. This is found in the animal cells and prokaryotic cells. Usually the cilia is in the animal cells, and the prokaryotic cells have the flagella. The plasma membrane is a flexible bilayer, phospholipid bilayer. It is the boundary for what comes in and out of the cells. That's why we call it selectively permeable. It also has transport proteins in them. We call the ones that are on the outside, just located on the outside or the inside, do not go through the whole thing, peripheral proteins. We call the proteins that go all the way through, that help with cellular transport, integral proteins such as channel proteins, carrier proteins, and some pumps. The plasma membrane can actually be found in an animal cell, a plant cell, and a prokaryotic cell. Ribosomes are organelles that help manufacture proteins. They're made up of two components, RNA and proteins together. This is where RNA translation happens after it has come from the nucleus, the RNA strand, the mRNA strand, the messenger RNA, as it was transcribed or through RNA transcription in the nucleus, comes out to the cytoplasm where the ribosomes are. This is where our protein synthesis happens, whether it be out in the cytoplasm or in the rough ER. So the lysosomes, only found in animal cells, are a vesicle, basically an organelle that contains things that can help, substances that can help digest excess or worn out organelles or food particles. When we bring in vesicles into the cell, sometimes we have to break down the walls to get to that solid particle or that liquid particle that we bring in. So lysosomes help with that. Vacuoles can be found in both animal and plant cells. Predominantly, when we think of vacuole, we think of the plant cell with the large central vacuole. You can kind of see it there in the image. It is a membrane-bound vesicle, and it's used for temporary storage of materials. In the plant cell, though, it helps to create the turgid pressure or the turgor pressure, which allows a plant to stand upright and so it won't wilt. So if that vacuole is full, it's going to be strong and it's going to continue to swell and stand upright because, again, remember, our plants don't have skeletons like us. In the animal cell, it's primarily used for storage of waste. Cell wall, only located in the plant cell and the prokaryotic cell. Very thick, rigid mesh of fibers, and it's on the outside of the plasma membrane. It helps with protection and gives it a little bit of structure. Cytoplasm is the semi-fluid material, mainly composed of water, and some people like to call it jelly-like, that the organelles are suspended in. So it's kind of the goo that everything sits in. It's found in all three, animal, plant, and prokaryotic cell. Some big differences right here between the prokaryotic cells and the eukaryotic cells. Big things you can see right away. The prokaryotic cell tends to be very basic, tends to be your bacteria, unicellular organisms. They do have a central area where their DNA is located. It kind of looks like that mess of spaghetti there in the pill-like prokaryotic cell. It does have a cell membrane, cell wall, and it does have ribosomes. And it has the flagella there for movement. When you move into the animal and the plant cells, you can kind of see they're more detailed, have a little bit more organelles to help with the structure. And it just has some different things for the animal cell and the plant cell to be able to carry out its processes. Has a lot more organelles, more detail, more functions. A big difference between the plant cell and the animal cell is size. You can kind of see that the plant cell is a lot larger than the animal cell, and it has that large central vacuole. So what else we need to talk about here on this little one? This this lecture 2 is cell theory. So we'll kind of go over cell theory a little bit. Remember, cell theory had a lot of different scientists involved. We have Hooke. I'll pull this picture up real quick so you guys can see. This is the one we used in our Pear Deck. You have Hooke, who was the first one to look at cells through a microscope. Okay, he was the one that decided to call them cells because they looked like the cells of the monastery monks that they were living in at the time. Remember, Leeuwenhoek was the one that created. He was the first to create a microscope, actually, a more powerful microscope than what Hooke was looking through. And he was actually looking at microorganisms in the pond water. Then we had Schleiden and Schwann that looked at some plant cells, and they were looking at different cells and understanding that's where that cells made up our plant cells, and then our plant organisms. And Virchow was kind of along the same lines. He was looking at animal cells in the same way. If you look at this image here, this is the 3 points of cell theory that are really important. All living things are composed of cells. So when we looked at that, we're looking at either unicellular or multicellular organisms. Cells are the basic units and structures and functions of living things. Got to have cells put together to make more complex organisms. And then all cells are produced from other cells. So we have that whole idea, kind of like what Virchow went to at the end, that we all come from more cells reproducing, replacing worn out cells, or creating new organisms. So this will be on your quiz on Friday. You need to go ahead and make sure that you stay on track with your studying of cell parts. Make sure that you look over that along with the cell transport.